In our previous lesson from Chapter 10, we talked about the concept of sexual reproduction and how important variation is. We also talked about the differences between meiotic and mitotic cell division. Today what we're going to be looking at are the events of meiosis, the specific details that make it different from mitosis. We're going to put these into a few categories. The first key event is the pairing up of homologous chromosomes. If you remember, we talked about diploid cells are cells that have homologous pairs in them. Well, in meiosis, we're going to see these homologous pairs all forming together, where each chromosome finds its partner chromosome, the one that looks like it, has the same size and shape, and has genes for the same traits on it, although the two would not be genetically identical. When they pair up, we call the process synapsis. Now, we also said in our previous notes from Chapter 10 that in meiosis we have two divisions of the nucleus, meiosis 1 and meiosis 2. Well, each one of those divisions has the same stages that we saw in mitosis: prophase, metaphase, anaphase, and telophase. Well, this synapsis process takes place during prophase 1. And what we see are two chromosomes, each one of which is already replicated. So each chromosome has two chromatids. Shown here, we've got the blue one on the left and one on the right. We've got a purple one on the left and on the right. These two chromosomes have found each other, and these two chromosomes look about the same. They're about the same size and shape. They, as we said before, carry genes for the same traits, but they're not genetically identical. This blue chromosome perhaps maybe came from this individual's father, and the purple one maybe came from his mother. Now, in real life, they would not have these actual colors, just so we're all clear. We're just doing that so we can tell them apart. Well, when we have the synapsis process take place for all the homologous pairs, each pairing is referred to as a tetrad. Now, the prefix tetra means four, and so in this tetrad, what there are four of are four chromatids. The blue chromosome has two chromatids. The purple one has two chromatids. The reason that's important is eventually these two chromosomes will be split into a total of 4 cells at the end of meiosis. Each cell will get exactly one chromatid from the tetrad. Now, also during this process of synapsis, when the two homologous chromosomes come together in each pair, the two chromosomes get so close, and because these are molecules that are so similar to each other, the molecules can swap parts. It seems kind of weird to us, but I always kind of think of it as if you were holding hands with someone, and when you went to let go, you ended up with their hand and they ended up with yours. Well, because chromosomes are molecules, this can happen, and it doesn't really affect them, but it does affect the results of meiosis. So this is why we discuss it, because we now have, for instance here, this light orange chromatid that has the dark reddish orange area swapped for what normally was the bottom of this. So this chromatid now is a mixture of some of the paternal information from this individual's father, and some maternal information, some from his mother. So as we said, since each chromatid ends up in a separate cell, we'll have from this one particular pairing the possibility of a chromosome that is all paternal, one that is half paternal and half maternal, a different part paternal, part maternal, and then one that's all maternal. Now, a few years ago when we used to discuss crossing over, we would say this is something that would occasionally happen. But the more we study it, the more we're finding out this seems to be kind of the norm. This happens frequently. There may be several locations of crossing over, even along the same chromatid pairing. And the end result of this is getting even more variation than what we had before. Simply creating some cells with all maternal and some with all paternal genes would create some variation, but that mixture, the combination, is going to give us even more than what we would see otherwise. The second key event in meiosis is going to be when those homologous pairs then separate. And of course, we didn't see this in mitosis because in mitosis, the homologous pairs never formed up to begin with. So this takes place right at the end of metaphase 1, because in metaphase 1, instead of seeing chromosomes lined up along the equator, we have tetrads lined up along the equator. And so we have the 4 chromatids in a set, and they're going to separate. But in metaphase 1, they're lined up. In anaphase 1, they've now moved apart, and we're seeing an entire replicated chromosome being pulled toward one pole, and its partner replicated chromosome being pulled toward another. Notice each chromosome still has its copy attached, although the copies have now been altered by the crossing over that we saw in the previous slide. Now, what's going to end up happening here is we're going to see a new nucleus forming that has a set of 3 chromosomes. In this particular diagram, I'll move this down here so we can see it better. In this diagram, we had 6 chromosomes to start with. Well, we're now going to have 3 chromosomes in each nucleus. And what we can see here is in the chromosomes, this mixture of, again, paternal and maternal genes that are on there. And we're eventually going to form 2 complete nuclei out of this. There wouldn't be any overlap there, but the size that I made my circles there, we see a little overlap. Each one's going to have 3 chromosomes at the end of this step. So what we have then at the end is 2 haploid cells, because we started with 6 chromosomes, we're down to 3. We started with cells that had pairs, now the cells won't have pairs. And at the end of meiosis 1, those 2 haploid cells are each a mixture of genes that came from this individual's mother or father. The third key event in meiosis is those chromatids that are copies of each other need to be separated from each other. Remember that during interphase, each chromatid made a copy of itself. Well, now those need to be separated. And this looks very similar to what we saw in mitosis. In fact, all of meiosis 2 looks extremely similar to mitosis, the only difference being the number of chromosomes. If this is a human cell we're looking at, in mitosis we're going from 46 chromosomes in the original cell to 46 in the daughter cells. With it being meiosis, in the beginning of meiosis 1 we had 46 chromosomes, but at the end of meiosis 1 we already were down to 23 chromosomes. So in meiosis 2 for a human, you start with 23 chromosomes, you end with 23. Now in anaphase 2, we end up seeing these chromatids splitting from their copies, but they're no longer true copies because of crossing over. It's no longer a true copy that we're looking at here. But we're going to end up seeing a place where we have 4 separate nuclei being formed. And we're going to, again, take a look at these. We'll kind of draw little ovals over them to represent the new nuclei that we'll get. And each nucleus is going to have still 3 chromosomes like we saw at the end of meiosis 1, but each chromosome no longer has a copy attached to itself. The chromosomes are now single chromatided chromosomes. Now, if we're talking about animals here, the whole point of all of meiosis is all about forming gametes, whether it's forming sperm or egg. And the process of forming gametes has the name given to it of gametogenesis. The prefix gamete, very obvious there, but the ending ogenesis, the term genesis is usually used to describe either the beginning of something, the formation or creation of something. So we put those 2 ideas together for the formation of gametes. And again, if we're talking about what's going on in animals, this is part of an overall life cycle. Just like we saw in chapter 9, where we saw the cell cycle, what was kind of like the life of a cell from the time it was formed till the time it was done. We're going to look at the same thing for an entire animal here, and what its life cycle is in terms of reproduction. Now, this is not the same as the life of the animal. It's just the life cycle of the animal. Being a cycle, we can start at any point. So let's start with gamete formation, since that's what we're talking about here. So at some point in an animal's lifetime, it's going to be making gametes, and it's going to make gametes by the process of meiosis. Well, the whole point of making gametes is that those gametes can fuse together with other gametes by the process of fertilization. So a sperm and an egg fuse together, they make a zygote, that's the fertilization process. Well, what's that zygote going to do? The zygote is going to grow up, it's going to go through development, it's going to use primarily mitosis to now become a multicellular large organism that looks like its parent. And once it is fully developed and sexually developed, it's going to start the process of gamete formation for its generation, and the process will continue on and on. So again, we have a life cycle. Well, if we look at just the gamete formation portion for animals, It does vary depending on whether you're talking about males or females. We'll start with the males first because it's a simpler idea here. Gametogenesis for males is specifically called spermatogenesis, and we start with a cell called a germ cell. It's short for the idea of germination, meaning to produce offspring. Now, a germ cell is diploid. It has pairs of chromosomes, and in this particular diagram, we're showing a cell with 4 chromosomes. Now, those germ cells go through mitosis like other normal cells in the body of an organism, but certain cells they'll form are referred to as spermatogonia is the plural, or spermatogonium is the singular. We have one spermatogonium here, still with 4 chromosomes, still diploid, because it went through mitosis, which maintains chromosome number. The spermatogonium goes through mitosis to make cells that are called primary spermatocytes. Again, still diploid. Now, primary spermatocyte, the name itself, primary means the first, and cyte is a cell. So this is the first step truly along the way to being a cell, a sperm cell. And it's going to go through 2 meiotic divisions. In the first meiotic division, it's going to form 2 cells, each of which is haploid. And those cells are called secondary spermatocytes. And we can see we've now reduced the chromosome number from 4 in the original cell down to 2, but each chromosome has a copy attached. It goes then through meiosis 2, the second meiotic division. We get a total of 4 cells. Each cell has now 2 chromatids. So in the second division, we've gone from haploid to haploid. These cells are referred to as spermatids, and a spermatid will then mature and change into eventually a sperm cell, which is still haploid. The key difference between a spermatid and a sperm cell, the sperm cell is now ready to swim with its flagellum attached. So just kind of giving names to the different stages that we saw as we talked through meiosis. And we're now going to look at females. The names are about the same, but there is a key difference in how this takes place. Now, it's not called eggogenesis, unfortunately. The prefix meaning egg is O, just like we see in ovary and in ovulation. So we went the prefix O and the ending ogenesis, so it's referred to as oogenesis. It's tempting to look at it and call it oogenesis, but that's a mispronunciation there. Now the words start out the same here. We start with a female germ cell, which is diploid, going through mitosis to become an oogonium, still diploid. That goes through mitosis to become a primary spermatocyte, still diploid. In the first meiotic division, we get haploid cells, but we don't get 2 secondary oocytes, which we would have expected if you remembered what happened in spermatogenesis. We get 1 secondary oocyte, and we get 1 very, very small cell that's called a polar body. Now, the difference between the secondary oocyte and the polar body is all about cytoplasm. During this division of cytokinesis going along after the first meiotic division, all of the cytoplasm moves into 1 cell, the secondary oocyte. So the first polar body is pretty much just a nucleus within a cell membrane wrapped around it. There's not enough cytoplasmic material for this cell to survive. But basically what's happening is the primary oocyte was kind of casting off excess genetic material that it couldn't use. Now, an egg cell needs to be very large. A sperm cell has very, very little cytoplasm. And so during fertilization, the egg cell is going to provide all of the cytoplasm for what will become the zygote. So it's going to be large. It's going to now steal basically all the cytoplasm from its sister cell that was formed. Now, each of these 2 cells will go through the second meiotic division. The secondary oocyte will become an ootid, and the ootid will also then produce a polar body off the side of that. The original first polar body will split into 2 more polar bodies. If it doesn't disintegrate first, it may break down before it fully finishes that process. Because what happens is all of these polar bodies, without having enough cytoplasmic material, are going to simply break down and not be functional cells. The ootid will continue to grow and mature and will become an ovum, which is the true egg cell now for this female. That was all in animals. I'm going to save you a lot of worries and a lot of headaches right now and say we're not going to look at plants at this point. The plant life cycle is very different, very bizarre, nothing whatsoever like what we're used to when we think of how animals reproduce. And it's not the reproduction process that's strange, it's actually the whole concept of what a generation is in plants, and the fact that the chromosome number in plants alternates generations, where one generation is diploid and the next one is haploid, and then back to diploid and so on back and forth. Very bizarre to us because it's not at all like the way we reproduce. You can take a look at it in Chapter 10 in your textbook, or if you don't want to dig into all those details right now, I promise we will talk about it later on in the year when we talk about plants. But that is the end of all of our notes for Chapter 10. Please ask your questions in class, and have a great day.